everyone, and welcome to this Integrated DNA Technologies webinar on our gene synthesis technology and an applications update. My name is Dr. Hans Packer, and I'll be serving as the moderator for today's presentation. The presentation today will be given by Dr. Adam Clore. Adam manages our synthetic biology design and support group here at IDT, where he's also actively involved in product development, research, and biosecurity. Adam has a background in microbiology and biochemistry, and he earned his PhD in biology and systems science from Portland State University, where he studied the molecular genetics of hyperthermophilic archaea and their viruses. Adam's presentation today should last about 30 minutes, and following that presentation we should have plenty of time to answer questions from attendees. So if you have a question about, whether something we have is applicable to you, you can ask those questions at that time. As attendees, you've been muted, but we encourage you to ask your questions or make comments anytime during or after the presentation, and you can do that by typing it into the questions box, which is located at the right-hand side of your screen in the GoToWebinar control panel. And if you go about halfway down, there's a thing that says questions with a plus sign or a up arrow, depending on whether you're on a Mac or a PC, and if you click on that, it'll make the window larger and you can type your question into that. At the end of the presentation, then I will direct questions to Adam, so we'll get through as many of those as we can. Also, in case you need to leave early today or you want to revisit this webinar later, we are recording the presentation, and we will make that available on our website, which is www.idtdna.com, and you'll find our video library under our Support and Education tab from the homepage. We also have a YouTube channel where this and other videos will be posted. There's quite a bit of content there, and that is www.youtube.com/IDTDNABio. We also will be posting the slides for this shortly, and the slides will be posted at www.slideshare.net/IDTDNA. So with those housekeeping things out of the way here, I'm going to let Adam get started with his presentation. Adam?
All right. Thanks, Hans, and thanks to everyone for attending today. I want to start out just going through and kind of giving an overview of our synthetic biology group, the products that we offer, and some of the ways you can use our products. I'll go through, particularly with a focus on G-Blocks gene fragments, some of the uses that have developed, especially some of the newer uses that came up in the past year or so. And then after that, I'm going to run through some of the new enhancements that we have to our products, to the ordering system, and to some of the design options that we have available. And then we'll wrap up talking about some new products that are coming down the pipeline. So to start out with, we'll talk about G-Blocks gene fragments. This is a product that we developed here at IDT about 3 years ago, and this at the time was the first commercially available synthetic linear double-stranded DNA fragment that you could purchase. At the time, we started out with a fragment that was 500 bases and up to 500 bases in length, and started out as a very high-fidelity sequence-verified product. And we've continued to make that product over the years, and we've expanded the capabilities of it. So we now have lengths that come up to 2 kb in size. We've increased our yields up to 1 microgram for our larger fragments. We've added libraries, and we've been able to decrease the delivery time to have a median shipping time of just 3 business days for our G-Blocks. So we've made some advancements with it, but we've kept a lot of what makes this product great, which is the consistent quality, the low price, and the fast turnaround time. So to look at some of the measures of quality here, you can see what the G-Blocks proportion of 100% correct sequences is across the range of sequence lengths here. This is looking at 262 G-Blocks that were sequenced on our Illumina MiSeq next-gen sequencer. What you can see here is that, pretty consistently across all of the lengths here, we have 80 to 90% of the sequences that are 100% correct. And of those sequences that are not 100% correct, most of those sequences contain a single base error or a single base deletion. So again, very minor changes in a minor population of what is mostly mostly correct products. So this is very similar to what you see, in PCR amplification from high-fidelity polymerases. So again, a product that is very high quality, very consistent, and again has a fast turnaround time. So let's jump in and talk about some applications of G-blocks. So we have a variety of different applications that have developed over time. And a lot of these applications have been described very well in the many citations that we have available for G-blocks. We have over 100 citations available on the website, and I believe that the number's approaching 200 now of total citations within the literature using G-blocks. And this has went way beyond the original use that we had envisioned for G-blocks, which was that of gene construction. And I'll go in through some of these other uses and describe some of those in the next few slides here. So we can start out with gene construction. This was the original idea with G-blocks gene fragments. These would be fragments of genes that could be put together, stitched together, and used to build large constructs, and that's what a lot of people use these for. Not just genes, but entire gene pathways, full viral genomes in some cases, partial viral genomes for vaccines, of course individual genes for protein evolution, and variants of genes can be built all with G-blocks. And this has some real benefits compared to a lot of traditional methods of either PCR amplifying out sequences that are desired, or cloning or using some other methods of doing this. When you use G-blocks gene fragments, you can get the sequence that you want, and you can go ahead and just clone that directly. That sequence doesn't have to be anything occurring in nature, so it doesn't limit you to the possibilities that are already available and could be PCR amplified. We have a very large acceptance criteria. We can't accept every sequence that's envisioned, but we have a very large acceptance criteria for sequences, and again we have that consistent turnaround time and a minimum of delays. We also have a synthetic biology design group that is dedicated to assisting customers in projects and helping get those projects into the pipeline. So we can do things like codon optimization. We can split large constructs into individual G-blocks that can be assembled using a variety of different methods, including many of the isothermal assembly methods, so like the Gibson assembly method, or SLIC, RIC, Golden Gate, any of these types of methods. And we can help track orders and make the arrival of those orders as predictable as possible. So to move on, one of the newer applications that G-blocks have been used for, and something that quite honestly we had never thought of, was using G-blocks as controls for NGS. For next generation sequencing, one of the technologies that's really taken off in the last few years are technologies that allow you to enrich for certain sequences or pull down certain sequences. And this type of enrichment can really focus the sequencing power of next-gen sequencing on specific and needed sequences. And what we can offer are controls for those types of enrichment. They can be spiked in as a G-block, and what that spike in can do is act as a control that can tell you your levels of enrichment. It can give you an idea of where you need to optimize things. It can help you along in that process. We also make G-blocks for customers to use in sample tracking as a spike-in. By doing this, by spiking in samples, G-blocks into samples after they've been collected, you have an internal identifier that can identify that sequence no matter whether a label is lost or a tube is mixed up or anything happens to it. So it's a great level of security to have available. Again, the G-blocks are really useful for this because they have no plasmid backbone in them, and because they are made completely synthetically, they have no genomic DNA contamination in them. So it's a really nice, really clean way to put a control into something. Again, flexible design, and those design options can be explained in detail by the design team here. They can be contacted at genes@idtdna.com. Okay. One of the applications that gBlocks has been used for a long time is the creation of variable domains in antibodies. Our original gBlocks, as I mentioned, had a 500 base limit, and that fell really, really nicely into the light and heavy chain variable regions for antibodies. So we had a lot of large orders early on, and we continue to this day to have a lot of large orders for these variable domains. gBlocks make a great are a great option when you're designing these types of variable regions. You can clone those into antibody expression vectors, and that's been used for anything from CAR T cell therapy to antibody-based vaccines and different therapies that are antibody-based, and even creation of simple detection antibodies. Basically, anytime that you need to create new variation or optimize binding efficiency with the antibodies, gBlocks can help by creating those variable domains. We can create large numbers of sequences, particularly when we use gBlocks libraries, which can incorporate random bases and random codons in the form of NNK bases at various sites within the variable region. It allows you to create thousands, and in some cases millions of variants of antibodies at a fairly low price. We can make these in high throughput, so we can make specific sequences in plates, and we can help with library design, plate orders, and sequence optimization of all of these processes. Another application of gBlocks, and one that is quite popular, particularly in the diagnostics and detection space, is that of a PCR control. And we design a wide range of PCR controls made from gBlocks. I think one of the real advantages really comes in when you're talking about using controls for multiplex PCR. And the reason for that is using a gBlock, you can put multiple templates, multiple targets on a single gBlock. By doing so, you ensure that you always have an equimolar ratio of the different targets on the, when you spike that into an assay or when you use that to build a standard curve. So it's a really nice control to have in the experiment there. Again, there's a very low level of cross-contamination. We often see, if there is any detectable contamination, it's often 20, sometimes even 30 CQ below the limit of the actual intended target that's present. So very low detection. We can do things like custom formulation, and we can also help with design, including buffer sequences in there that can be handy to detect things, and they can help in manufacturing. And we also offer a gene service that allows you to move those sequences into a GMP space. So we can make the GMP genes that can take a diagnostic control all the way through the process for you. So it's a nice full spectrum order here, and we can get any of that started and answer any questions again through the email link genes@idtdna.com. So one of the neatest tools I think that came about in the last couple of years is CRISPR. And I'm sure most of you have heard of CRISPR. This is a technology that is used to create targeted double-stranded breaks within a genome of a wide variety of organisms. And gBlocks have been at the forefront of CRISPR technology really since the inception of it. Some of the early papers that came out in 2013 from the Church Lab cited using gBlocks to create the gRNA sequences that were cloned into some of the plasmids that are used to express gRNAs and to target specific sites for the double-stranded break. And that's still used to this day. We have a lot of customers that order gBlocks to be cloned in or to be used just directly in cells. We have protocols available that allow you to transfect a direct linear gBlock into a cell. That gBlock contains a U6 promoter on it and can transiently express the gRNA long enough to create a CRISPR-mediated break, and then afterwards is degraded because there's no ability to replicate within the cell. And so you have a transient infection that benefits from not having that gRNA floating around after the transfection happens. We also have a new non-gBlock product that is great for CRISPR, and this is called the Alt-R product. These are chemically modified synthetic RNAs that mimic the natural CRISPR RNA and tracer RNA and can be used in combination with our Cas9 protein and transfected into cells to create a very efficient double-stranded break. And so there's two technologies that are here that can be used to create this break. But beyond just creating the double-stranded break, we want to think about being able to repair that break. And naturally, the way cells will repair that break is to use the non-homologous end-joining repair pathway, which is an error-prone repair pathway and repairs the break by creating a small insertion or deletion called an indel in the area of the break. And that's great if you want to disrupt the activity of a gene. The indel often creates a frame shift, and you can very easily disrupt activities of genes. But oftentimes you want to actually replace the activity of the gene, modify the activity of the gene, or add additional genetic information into the targeted break site. And to do that, you need to take advantage of the homologous recombination system that is present in cells. This is a system that can combine or can recombine DNA, and by creating fragments of DNA known as donor DNA, this is DNA with two homology arms that flank a double-stranded break. You can use the homology-mediated repair system to insert that piece of donor DNA. And if you put a sequence of DNA that is desired between those homology arms, this insertion reaction will insert that targeted locus in the place of the double-stranded break. That targeted locus could be as small as a SNP, maybe that changes the existing SNP into a different sequence. It could also be something larger, something that could encode an entire gene or even a cassette that may contain something like an antibiotic resistance or a GFP marker or a wide variety of different things. So we can help design these as G-blocks, gene fragments, or as genes, depending on the application and depending on the size that you guys are looking at. Again, the group at genes@IDTDNA.com is happy to help with these things. We can help design these, and we can design these in concert with designs of the alt-R synthetic RNA that can be used for the actual targeting. So there's a lot of different options there. It's a really exciting field, and if you have any questions, we'd love to talk to you about that. One of the other things that I want to talk about is not just an application, but a community that we're really helping to support here, and that's the IGEM teams. This is something that, this competition is something that IDT has sponsored for the last 4 years, and it's an international competition of students that are at a high school level, undergraduate level, and even a graduate level, who all come together to do research and create projects and create new ways to use synthetic biology and use those projects in a competition that occurs every year in Boston. It's a very exciting competition. It is amazing what some of these students can do in a year, some of the projects that are developed, and there is just a wealth of exciting information and knowledge that's available there. And all of this can be found at the IGEM website, which you can get to through the link at IDTDNA.com/IGEM. And I would encourage everyone to stop and look this up. There's some really interesting things there. And Randy Retberg, the president and founder of the IGEM Foundation, I think is really someone whose vision has taken this competition and really made it what it is today. Really brings a lot of excitement to it. And there's a lot of information there and some. And you haven't seen this yet, IDT is offering 20 kilobases of free DNA to every IGEM team. And so we would encourage all of you to get in contact with us if you haven't already. If you're in the IGEM competition, we would love to help you out. And again, we'd love to support this competition. Okay, I want to switch gears a little bit now and talk about some of the new tools that we've developed to help researchers. One of the things that I mentioned earlier on in the talk is that we can't create every sequence of DNA that everyone wants. And the reasons for that have to do with the structure of DNA itself and just limitations to synthesis. There's certain things like high GC content, repeat density, and secondary structure that can complicate synthesis. They can also complicate things like cloning and a lot of downstream applications. And so to eliminate sequences that we're going to have a hard time making and that our customers are going to have a hard time using, we've created a set of rules that can identify structural complexities. And we've also created an online tool that can show you within your sequences in real time where those structural complexities lie, what those structural complexities are, and give you tools to help fix those or help change those so you can minimize the complexities and get your sequences ordered. So this is an example of what the tool looks like. There's a rule violation here. There's a large repeat sequence that is present here. It identifies where the repeat sequence is, and it allows you in this window to edit the sequence. You can also do things like codon optimize the sequence. The codon optimization can automatically detect start and stop codons. You can also manually select reading frames and the locations or subsections that you want codon optimized, and you can select from a wide variety of different organisms to optimize the codons too. So it's a nice, easy tool, and it's something that we hope allows customers to see what complexities are present in their sequences and rapidly find a way to modify those sequences to help eliminate some of those complexities. Of course, our tools that are online are not the only way that we can do this. You're always free to call us or email us at IDT, and we can help you. We have a large team of design personnel that can help with design questions and optimization tools. We also have some videos that are available on YouTube that explain some of the complexities, some of the rules, and also some of the codon optimization parameters that can be used. And just to jump into the codon optimization a little more, we have that built into our online rules tester that allows you to fix it. We also have a separate standalone codon optimization tool that allows you to enter sequences either individually or in bulk, in DNA bases or amino acid sequences, and they can optimize for, again, a wide variety of different organisms, and they can optimize them and screen them to find the best optimization in the GBlocks rule set or in the genes rule set. So either one of those can be used. These are slightly different rule sets. The gene fragments have a slightly different set of values than the genes. So we can optimize for either one of those depending on which product you would like to finally use. Again, you can set the reading frames however you like. There's an auto detection, there's a manual selection where you can paste, and you can also identify things by base number. So however you wish to modify these, you can go ahead and do that. Again, there are tutorials available at YouTube that can show you how to use this tool, and we are always happy to help answer questions with this. One of the other tools that we've recently brought online is the order status page. So this is a tool that allows you to see the current orders and the past orders in the synthetic biology group that you've had at IDT. So any genes or any GBlocks gene fragments will show up in this. It gives you the progress, the state of progress of any of the individual sequences, and an estimation of ship dates or an actual ship date if that item has actually already shipped. So this is an example of what you can see is a selection of sequences that are present in a sales order. These all have an actual ship date instead of an estimated ship date because these have all shipped. You can see they've all completed. The green bar has moved all the way across and they've all completed all steps. And at this stage, you can go through and you can download any of the documentation that goes along with this, spec sheet, sequencing data, different things like that. And you can also verify the progress of things that are not completed. Obviously, this is showing things that are completed, but for items that are not completed, what we have is this estimated ship date, which shows you our best estimation of when it's going to ship. And this estimated ship date is something that is updated dynamically. So as a product moves through the synthesis pathway, this estimated ship date goes down based on the average time to ship from whatever state in manufacturing or QC the item is at. So it gives you an accurate estimation of shipping. And in the cases where we do run into problems with something and we have to take a step backwards in manufacturing, this estimated ship date takes that into account and adjusts that shipping date backwards. So it gives you the most accurate estimate that we can give you on the time that you're going to receive the item. We know we can't always anticipate every single problem in custom DNA synthesis, but our goal here is to make it as transparent as possible so that you can predict and plan around when you're going to receive your items. One of the other really exciting things that we've seen recently is the incorporation of NGS analysis in our genes. So we've started using NGS analysis for genes larger than 500 bases. And the reason for this is it gives a very high depth of coverage of the genes, and that's really nice for a couple of reasons. One is it allows us to see if there's any minor populations within a sequence. Now, all of our gene sequences are cloned and grown from a clonal population, but particularly in sequences that may have some toxicity or some instability, those clonal populations are not always 100% the same. There's some heterogeneity sometimes within those sequences, and we can identify that using NGS analysis to a level that we couldn't with traditional Sanger sequencing. We also, because we have very detailed information about errors that occur within sequencing, we can use that back into our rules engine, and we can use that information to make our rules engine better. And we're using that to increase the predictive accuracy of the rules engine, which in turn gives you a better estimate of what we're going to be able to make, allows us to eliminate delays, and make sure that you get the product you need on time. And this is just an example of an imperfect sequence. Obviously, this has a sequence that has one base that is completely wrong in it. It's very easy to identify and very easy to see, particularly with the depth of coverage that we have here. The improved rules engine is something that we're working on constantly, and this is something that we're updating a lot. And this is just an example of a single rule that we look at and the successes and failures that occur with a test set of sequences as the complexity of that rule becomes larger and larger. And so this allows us to identify exactly where the problems lie within a sequence, and again, predict the increase the predictive accuracy of the scoring and the rules that we're using. All right, I want to mention just really quickly, we have in beta testing right now a new product that is that we're very excited about and we're interested in, and that is single-stranded G blocks. And these are particularly gene fragments that are going to be used for homology-mediated repair. As I mentioned a few slides ago, homology-mediated repair can insert DNA at the site of a double-stranded break, and single-stranded DNA has a distinct advantage in this respect, in that it recombines much more efficiently than double-stranded DNA, and that efficiency means that you can decrease the length of the homology arms that are needed on each end, and thereby shrinking the total construct size, often by over a kilobase. And by doing that, you make a smaller, easier to make construct. You can insert larger cassettes within there. It's something that seems to work very well. This is something that we're in a very limited beta testing environment right now, but it's something that we hope to release by the end of this year, and it's going to initially be a product that is up to 2.5 kilobases in size. If you have any questions about this, I would encourage you to email us at genes@idtdna.com. I can't guarantee that we can get you in a beta test for this right now, but we're happy to get you on the list, and we may be able to work you into some of the beta testing here in the near future. Okay, with that, I just wanted to touch on a little bit of the custom design support that we offer for G-Blocks. And it's hard to explain a lot of the support because we do such a wide range of different types of support. And so this is just a list of a few different types of support or custom requests that we've helped design in the past few years. One of them was a rather large sequence that contained no C bases on one strand and no G bases on the other strand, but had relatively balanced GC content over the entire sequence. Another option is, or another design request that we fulfilled was to generate unique code tables based on sequencing data. So this is for an organism that it was not represented in the sequencing database and didn't have codon tables derived for it yet, so we're able to help derive those and design codons to match that usage. We've also done things like minimize GPC dinucleotides while maximizing GC content in third positions of codons. Again, these are just a very small subset of the wide range of designs that we do for customers. And if you have any more questions about that, I would encourage everyone to email us at genes@idtdna.com, and we can provide you with more information. Lastly, I just want to take a minute and talk about biosecurity and the importance of biosecurity here at IDT. IDT was one of the founding members of the International Gene Synthesis Consortium, which is a non-for-profit corporation set up solely to help guide and help monitor screening of gene sequences and synthetic constructs to make sure that they're getting in the hands of researchers who are doing good research and keeping them out of the hands of anyone who could use those for harm. So we've set up what we've called the Harmonized Screening Protocol, and this protocol is something that is a guideline followed by almost all of the gene synthesis companies worldwide, at least the major gene synthesis companies worldwide. And it's a protocol to screen both customers and sequences to verify that these sequences are going to be used for good and that these are going to be safe sequences to manufacture. It's something that we take incredibly seriously here at IDT, and it's something that we work with our competitors on to ensure that we're doing this in a safe and responsible manner. And if anyone has any questions about that, feel free to contact us at the website genesynthesisconsortium.org. We can get you in touch with the committee there. And for anyone working in academic institutions that would like to become a part of this community, we have that option available. And so I would again encourage you to go to the website. There's a lot of information there, and also some contact information for anyone interested in learning more about biosecurity and how we screen customers and screen sequences. Finally, I'll leave you guys with the additional resources that we have available. I've said several times now, I think that you can contact us at genes@idtdna.com whenever you have questions. We also have more product-specific information on gene fragments at idtdna.com/gblocks and idtdna.com/genes for any of the genes information. Lots of information here, protocols, citations, and different things like that. So something definitely worth checking out. And with that, I'll end the presentation and take any questions that anyone has.
Okay, thank you for that presentation, Adam. So at this time, if you haven't already done so, you can ask your questions by typing those into the questions box in the GoToWebinar control panel. So if you go about halfway down, there's a thing that says questions that has either a plus sign or an up arrow, depending on whether you're in a Mac or a PC. You can just expand that and type your question into the questions box. We do have a couple of them already, so I will just get started. The first thing that I was going to ask Adam was, can you just comment a little bit on where IDT is as far as the quality of the G-BLAST gene fragments compared to other people that offer similar products?
Sure, sure. There is quite a wide range of quality that's available in products like G-Blocks that are available. This is something, as we mentioned, that we pioneered about 3 years ago when we came out with G-Blocks gene fragments. And since then, there's been a lot of competition that has developed around this. IDT still leads the industry in the quality that we offer. There are also some of our competitors that are of very similar quality to us, quite honestly. So there's definitely a lot of competition out there. There's also a wide range. So there are some of our competitors that have fairly poor quality products and products that contain a lot of errors and deletions and fragments within them. So there is a wide range. I think the real benefit that IDT has, even over the competitors that have a similar quality product, is we're able to manufacture a very wide range of the sequence space. We can manufacture complexities that many of our competitors won't take on, and we can manufacture those consistently, and I think that's what we've really strived for, and what we've really achieved here at IDT is the ability to make these consistently. Quality is great, but if you can't get it in time to do the experiments that you need to do, we realize that this is one of the early steps in anyone's experiment, and if the early steps are unpredictable, the rest of the workflow suffers from it. And so we've really focused on getting the product made at high quality and in a very consistent turnaround time. Again, this is one of the reasons that we went ahead and developed our status page to allow customers to see, as transparently as possible where we are in our manufacturing process and how we can get the products out to the customers. So I think that's that's my overview of the current state of the competition.
Okay. Here's a CRISPR question for you. This person's asking, in your experience with the CRISPR-Cas gene replacement that you discussed, do you find it is better to use 2 guide RNAs? And then often I've seen only a single guide RNA to target the DNA you want to delete. So do you want to talk about that? And I think you could talk about the context between the alter CRISPR system versus...
Yeah. So I think what the question is really addressing is whether to use the naturally occurring dual-stranded nuclease that's present in the natural Cas9, or using 2 Cas9s that are modified to be nicases. By doing the latter, what you generally do is you target 2 sites very close to one another, where the nicase cuts each strand individually, and by doing so, you target a site much more accurately, because obviously you need 2 targeted events to occur simultaneously to create that double-stranded break. This has been shown to be much more accurate in some of the early papers that came out, and in some cases it was orders of magnitude more accurate than a single nuclease, a single targeted dual nuclease. That being said, almost everyone uses just the single targeted nuclease, and the reason for that is, even though the accuracy is higher with the dual nuclease, there's a lot more design challenges around it. It's a lot harder to get into the system, and it's not as reliable. And what I think most people have opted for is taking a slight hit in target accuracy for the ability to rapidly move through the process and cut down the design time and get through the process. The other thing I'll mention with that, it may be slightly less, and sometimes quite a bit less accurate, but it is still very accurate to use a single target, a single guide RNA and that seems to be able to get the job done and get the job done a lot quicker. So that's what most people are using, and that's what I would suggest to use.
So you touched on this already, but this person asked, what would you use the single-stranded DNA fragments for?
So single-stranded DNA fragments and double-stranded DNA fragments can both be used for homology-mediated repair. And we can jump back to the slide on homology-mediated repair. There we are. So this is a slide showing the donor DNA that's used in homology-mediated repair, and this is shown as double-stranded DNA. This donor DNA has these homology arms that allow the naturally occurring homologous recombination system to recombine this, and by doing so, after this recombination event happens, the target locus is recombined into the break and replaces the DNA within that break. So it's a good way to shuttle DNA into the site of a double-stranded break. Now, this works with both double-stranded and single-stranded DNA. With double-stranded DNA, particularly in mammalian systems, this is much less efficient than with single-stranded DNA. Oftentimes, these homology arms, when they're double-stranded, need to be hundreds of bases in length on each arm, whereas single-stranded stuff can be as short as 40 or 50 bases of homology on each arm. So the single-stranded option offers much more flexibility and is going to be a great tool once we have that publicly available. Again, we're just beta testing that right now, so we are somewhat limited in it, but we will have that out in production quite soon.
Yeah, and that being said, I mean, so that's a great application for that, but I can imagine that our customers will come up with some really cool other ways to use what is essentially a really, really long oligo. I'm thinking about like those DNA origami people, you know, like imagine the scaffold, you know.
Yep. Yeah. No. I have no doubt that there will be many more options. And you know, this is the same story that we saw with G-blocks themselves too. We built them for a very specific application, and since then the uses of it have just skyrocketed. It's been really amazing.
So it'd be cool to see what people do with that.
Absolutely.
So the next thing is, I'm actually going to read the statement before the question that's written in the reverse order. This person says, I work in bacterium which has a high GC content, and then 70% is the number in parentheses, but have generally avoided G-blocks gene fragments for, looks like knockouts, due to the problems associated with GC content. Does IDT expect to be able to synthesize more complex sequences in the future?
Well, I assume by more complex sequences that he's asking about higher GC content sequences, which are often less complex. That's one of the problems we run into with high GC content is you begin to have more, along with just high GC, you also tend to have more repeat sequences and things like that. And the answer to that is yes. We're working on that. We're working to relax the rules, particularly at the extremes of the GC content. Right now, our rule cutoff is 68%. So I can see why these are not a great option for him in an organism that has a 70% GC overall. We're going to relax that. It's not going to be a huge amount at first, but that is one of the areas that we are expanding into and working to access. So it's not going to be overnight. Our newest rules update is going to come out in September, and that will have some relaxation of the GC content, probably only by a couple of percent. So it's not going to be a major game changer, but it may get a few more things in that would help this individual.
Okay. This next question might be a little bit tough to address given the beta test nature of the single-stranded gene fragments, but can you comment on the sequence correctness and the stability of the single-stranded G-blocks gene fragments in comparison to ultramer oligonucleotides?
Yeah. So our sequence accuracy is going to be much higher than ultramers. So ultramers have an error rate that is one of the lowest error rates in the industry for synthesized DNA. That being said, it is simply the result of multiple couplings of bases together, and because those coupling efficiencies are not 100%, you have a chance of truncation and deletions at every base that's coupled. And so we have errors somewhere in the 1 to 300 range, and they can be even higher than that depending on sequence complexity. The sequences in our GBLOX gene fragments are much higher than that, and that's because even though we use ultramers to build these, the GBLOX go through a process that is proprietary to IDT, but which involves enrichment and elimination of deletions, substitutions, and errors. And so we actively are selecting for correct products. The single-stranded product that we make is going to be even more accurate than that, and I can't give a lot of details about that, but the accuracy is going to exceed that of our standard GBLOX. So it's going to be very accurate. As far as the stability goes, the stability of DNA, is fairly consistent across the board. Single-stranded DNA has slightly lower stability than double-stranded DNA, and that's because there is some base protection that happens in the double helix of the double-stranded DNA. That being said, we've done a lot of long-term stability assays on our single-stranded DNA in the oligo form, and those are quite stable in many periods, for up to years at a time. We expect nothing different from the single-stranded GBLOX.
Very related to that, Adam, can you say anything more about the stability of GBLOX gene fragments when they're dry versus in solution as far as stability?
Sure. Yeah, and this is kind of interesting. GBLOX themselves, again, are very stable. The bigger issue that we've ran into with GBLOX is that the long linear DNA tends to be sticky and tends to adhere to tubes. And so while we don't see a decrease because of degradation, we do see tube adherence from time to time. And that tube adherence is particularly present when the DNA is stored at low concentrations, particularly below 1 nanogram per microliter. The adherence can be pretty much eliminated with the use of a carrier molecule such as tRNA or even some synthetic oligos or anything like that. We have some protocols and some information available on that for anyone that's storing things at low concentrations or for a long time or both. So that's what I would recommend, for storage of GBLOX. As far as the actual stability of GBLOX goes, we have not done a long-term stability study with GBLOX, but we have used and routinely use GBLOX that have been stored for a long period of time, and we reconstitute things within the lab with good success. We've used them well over a year old, and we have customers that routinely use them for that length of time with no problem. Again, storing those, our most stable condition that we found when we've looked at storing things in pure water, storing things in TE, and storing things dry is actually storing them in TE. We were a little surprised at that. We thought that storing them dry would be as good or better than storing them in TE. But what we believe happens is even when you store them dry, there's a small amount of moisture in there that can actually act on the DNA, and you also go through the dry down process, which can increase oxidation and increase adduct products like adeoxoguanine. So storing it in TE and storing it at -20 or -80 is probably the best option for storing those. And again, if you're at a low concentration, storing that with a carrier in there is going to help for recovery of that material when you pull it back out of the storage container.
Okay, let's see here. So This person actually asked two questions. The first one was that they started off by asking they want to synthesize something that's 1,400 base pairs, and they want to know about online tools for optimizing that sequence. But the second part, they're asking about synthesizing something that's 1,400 base pairs that contains modifications.
Yeah. Well, so for the first part, it's a pretty easy answer. Our online tools can do some basic basic optimizations and things like that. I've discussed being able to codon optimize things, being able to manually edit things and edit things at the level of the codon. So you can do those through our tools, but we have no interest in trying to create a suite of tools. There's a lot of really great tools that are available out there. And we don't endorse any particular tool. We think you should find what works for you. But what I will say is that there are a few tools that really stand out as really nice products for us and things that our customers use regularly and work well. One of them is an online design tool by a company called Tesselligen. They make a nice suite of tools that can be used for design. There's a lot of codon optimization tools. There's some CRISPR design tools in there, so they have a very nice option. The guys at Benchling also have a great tool that's available. They also have an electronic lab notebook that you can use to store and collaborate with your colleagues. A lot of nice design options in there, including some CRISPR design tools as well. So there are a lot of options that are available, and we can address specific things with that with individuals if they have questions, if they just want to email us. But those are a couple of places to start with. Now, the second part of the question is a little harder. We have a very limited ability to put chemical modifications into our G blocks. We can basically only modify things at the ends of the G blocks, and even that we would have to do on a custom design. So it's something we haven't really done a lot of work with yet, but internal modifications are particularly hard for us because they interfere with a lot of the tools that we use to enrich and select for sequences that lack any errors in them. So that unfortunately is a limitation that we have with G blocks.
They should, I would assume that you would recommend that they contact you with the specifics of what they want to do, the genes at IDT DNA.
Yep, absolutely. And we've worked with a few customers in the past, and we've been able to come up with some fixes for things, but there's not
pretty limited.
Yeah, there's not a great solution, particularly if you want things in the middle of the sequence.
Okay. Here's a big picture kind of question. So do you imagine that the cost of synthetic DNA will continue to drop, or are we reaching a lower bound?
Yeah, no, that is an interesting question. I don't think that we have reached the lower bound yet, and I think there's some of our competitors out there that have some exciting technologies that are bringing in sort of large-scale gene synthesis into the realm of synthetic biology, and I think there's still some limitations with that type of technology, but I think that's a really interesting idea and an idea we're exploring as well. The other thing that we are continually doing is decreasing the size of the syntheses that we're doing. By doing so, we're decreasing the cost and the turnaround time. The real advantage, though, that we see of synthesizing things the way we do, and that is synthesizing individual items that can be individually QC'd, is it allows us to have a very fine control on the input that we're putting into the system and into the manufacturing process. And it's that input of being such high quality that allows us to get a final output that is of similarly very high quality and allows us to make a very nice and successful product like we've made.
Okay. I think that we've gotten through all of the technical questions here. For the people who ask CRISPR questions, we do also, I wish we had added that to the slide actually at the end. The www.idtdna.com/CRISPR URL will take you to our Alter CRISPR page. And not only is that a product page, but if you look, there's a lot of information on there about, how it works, and there's some data on the performance tab and stuff that you can really kind of delve into. And we've also got a wealth of videos and stuff in our genome editing section under our video library. So I'd really encourage you to look there to see like how what we offer compares to other technologies that are available. So yeah, www.idtdna.com/CRISPR. Yeah, and if anybody has any other questions, you can sneak them in really quick. Just really quick, this we are recording this, and this video will be available on our website in that video library, so www.idtdna.com, and then on the home page you can just look under the Support and Education tab, and you'll find our video library right there, and you can filter that down to whatever application you're interested in. Like I was just saying, genome editing is one of them. We also have a YouTube channel, www.youtube.com/IDTDNABio, and the slides will be available on www.slideshare.net/IDTDNA. And if you have any questions about any of that, you can contact us through the website, and we can follow up with you. All right, so with that being said, Adam, thank you very much for the presentation today, and thank you everybody for attending and the great questions. And yeah, we look forward to talking to you soon. We'll have other webinars in the near future. Adam, thanks.
All right, thank you, and thank everyone for attending.